Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to talk about a regulation of transcription initiation. But in this video we are actually going to talk about that how we can control the regulation of transcription initiation by a protein which we call that regulatory protein. In the previous video, if you guys remember, we talked about the induction and repression enzyme synthesis, that how it synthesized. And in that previous video we talked housekeeping function, we also talked housekeeping gene, we talked the beta galactosidase production, that how it is produced and how it perform their function. We also talked the constitutive genes, and we also talked inducible genes, repressor and repressor genes as well. So we talked each and everything in detail. So if you missed that video lecture, so go back to my channel and watch this video and then come to this video, inshallah. Then you will get idea about that how we can control the regulation of transcription by a regulatory protein. Okay, so let me clean the whiteboard and let me start. So now let me write regulatory proteins. What are actually regulatory proteins? These are the proteins which are produced by a regulatory genes. Okay, now we have inside the cell, if the transcription initiation process is carry on, so then we need regulatory proteins, and we call that repressor, we call that activator protein. Okay, so when the protein that are involved to turn on the genes, which we need, which the cell need, we call that regulatory protein, and that protein is actually called as activator protein. And there are some other regulatory proteins which are needed to stop the process of transcription. Okay, and we call that repressor proteins. Okay, so regulatory protein are repressor proteins and activator proteins. Now this repressor are mainly involved to induce transcription. This activator protein is actually involved to induce the transcription process, while this repressor protein are involved to repressed or inhibit the transcription process. Okay, so we have regulatory proteins in which we have repressor and activator proteins. Repressor protein are that regulatory protein that can inhibit the transcription process or stop the transcription process, and activator protein are that regulatory protein that can induce or enhance or start or activate the transcription process. So what does it mean? It means that this regulatory protein are actually perform two types of the function. One we call that positive, and other we call that negative control of transcription. So now this regulatory protein actually perform two type of the function. One we call it positive control of transcription, and the other we call it negative control of the transcription. Positive control of transcription means that process in which the activator proteins bind on the activator binding side of the DNA, and what it will do, it will start the process of the transcription. Okay, we call that positive control transcription. And negative control of transcription means that in this case we have regulatory protein which we call that repressor protein, which are involved to bind on a DNA or at the operator portion and to stop the process of the transcription. This is called as negative control of transcription. Then again another question that arises, that how actually the transcription is turned on and turned off, means how the transcription is activated and deactivated. These enzymes are actually involved in the activation and deactivation of the transcription. We have a DNA. Let's suppose this is a DNA, as you can see. So let's suppose this is a DNA. Inside this DNA, what we have a promoter region. Let's suppose this blue is the promoter region, P, and we have another region which we call that operator region. So let's suppose this is the operator region, O, and then we have some other region which we call that structural genes. Let's suppose this portion is called as structural genes, which are specially responsible to perform. These structural genes are actually involved to produce a particular type of enzyme. Okay, this whole thing is called as operon. Inshallah, we will explain operon in upcoming video lecture. Okay, now this promoter is actually the portion where this activator protein can bind. Okay, activator protein are bind at that particular portion and it will activate. Means when the activator proteins are bind, so then it will cause the bending of this DNA. DNA will bend. So when the DNA will bend, so then the polymerase enzyme, RNA polymerase enzyme will bend and it will start the process of transcription. So mainly the repressor, the activator protein are bound to the promoter region, and if the process, the transcription process which are need to be stopped, so then we have repressor proteins which binds to this operator region. So when it binds to this operator region, so what happen, the RNA polymerase will not bind, so then the process of the transcription will be stopped. So now what actually going on? Let me explain the whole process with a diagram. What's actually going on inside a cell? This is, let's suppose, the DNA, and this is, let's suppose, the operon portion where promoter, operator and all the genes are present. So at that particular portion, which is, this is actually structural genes which are involved to produce enzymes. Okay, now what's going on? Let's suppose this DNA need to be turned on, or these genes need to be turned on. So then what happened? We have the activator proteins. So this activator proteins will be look like in this way. Let's suppose this is the activator proteins. So what happened? These activator proteins are present, but still it is not in active form. Now to activate this activator protein, so what happened? We have our effector molecules, or which we call that inducer. So inducer, what actually it will do? It will activate the repressor proteins or enzymes. This repressor enzymes once activated, so then it will bind to this promoter region. Okay, so then it will bind to this promoter region. Now once it bind to this promoter region, what happen? The process of transcription will be start. It will cause the bending of the DNA. So when the DNA is bent, so then it will attract more activator and transcriptional factors are binded. So at that way, the RNA polymerase will be attracted, and this way the RNA polymerase will bind to this portion, as you can see. So let's suppose this RNA polymerase, it will bind and start the transcription process. But remember guys, if the transcription process which need to be stopped, so what happen in this case? This protein or this effector molecule is released out, which we call it inducer. The inducer will released out, and this enzyme will be in deactivated form. It will again deattach, means it will deattach from the promoter region. Once it deattach from the promoter region, so then the transcription process will be stopped. The DNA will again in a linear or in a circular form. In this way, the RNA polymerase will not bind and the transcription process will be stopped. This is the first way. This process is actually the first process, the way through which the transcription is stopped, we call that positive, and the second way through which the transcription is stopped, we call that negative. Second process. In the second process, what we have repressor protein. This is all about the activator protein, that how the activator protein are involved in the turning on of the genes. And let's suppose we have again the DNA, as you can see. O, and S means structural genes. Let's suppose all these things are again available. If we have a repressor protein, let's suppose repressor protein, so what happen? The repressor protein will bind to this area. Let's suppose this is a repressor protein. It will bind to the operator region, and once it bind to this operator region, so it will block the process of transcription. Okay, but remember guys, this repressor protein are not bind to this operator portion. It will bind at that time when it will connect, or when we have another effector molecule which we call it co-repressor. Co-repressor are actually the effector molecule that can bind to this portion, let's suppose to this portion of this repressor enzyme. When it binds to this portion of the repressor enzyme at the active site, so then what happen? It will bring some conformational changes, due to which it will bind to this operator region and stop the process of transcription. Okay, so if this co-repressor molecule are removed, so then the conformational changes occur in the repressor enzyme, and it will again deattach, and once it deattaches, so then the transcription process will be carry on. RNA polymerase will bind, or the activator protein will bind, and transcription process will be carry on. We called it positive. Again, we called it positive. And if it is repressor protein are involved, co-repressor are bind, so then this is called as negative transcription. Okay. Let me draw a third diagram. Sometime what happened? Let's suppose this is again a DNA, as you can see. Now then what happened? Sometime what happened? We have activator proteins. This activator protein doesn't need the inducer. Remember guys, sometimes we have the activator protein, it will bind to this operator region, promoter region. So when it will bind the promoter region, it will cause the bending of the DNA, and the transcription process will carry on, which we call a positive transcription control. So we get the product. But if we have inhibitor molecules, so this inhibitor molecules, let's suppose this is the process carry on, so then we have, if we want to stop this transcription process, so then we have another molecule which we call that inhibitor molecule. This inhibitor molecule binds to the active site. Let's suppose this is the active site. It binds to the active site to this site of the enzyme. So when it binds to the active site of the enzyme, so this activator protein, what happened? Conformational changes occur in the activator protein, due to which it will deattach. So when it will deattach, so again RNA polymerase are not bind. Why? Because the protein are not present, binding are not occur. So in this way the process is again stopped. Okay, so that's all about the control of transcription initiation by our regulatory proteins. That's all about the regulatory proteins and their function. I hope you guys get idea about that. If still you have any kind of question, you can ask through a comment. Inshallah, I will give you answer as soon as possible. Inshallah, in next video we will explain the operon concept, which is very important for you guys. Okay, so please stay tuned and keep watching. Thank you so much for watching that video.